Hello everyone, my name is Getsemani Arteaga, and today I'm going to be discussing my research over compound function value traits of Visia americana reproductive phenotypes. So to start off, I want to go ahead and talk about the plant-pollinator interactions. So the function of every flower is similar across the spectrum of different species and their interaction with pollinators. Animal pollinated plants aim to do two main things. They aim to attract pollinators to entice flower visitation, as well as promote efficient pollen transfer for reproduction between the flower and the pollinator. So when you look at these flowers, you will notice that not every single flower is identical, and when you look at these differences, it's not just random variation within the plant itself. Repeatedly produced flowers are not identical, as mentioned earlier, but they rather display systematic patterns of variation. For example, right here pictured is the flower delphinium, and you can see that the trends change as they go from top to bottom. So when you look at this measurement size of the floral length, you see how they decline systemically as you go from the lower bottom flowers to higher, smaller flowers. And these differences among these animal pollinated plants with repeated flowers are called architectural effects. Work from Sattler actually explains how these patterns of variation are maintained through what is called these architectural effects, which is dependent on for flower morphology and their position on the floral display. So these patterns of architectural effects can vary between individuals, and as you see here on the graph, you can see that the graph line or trend lines are not identical, but using this graph, you can imply that there is variation among the individual. So previous work from Kubaba et al. Has shown how these patterns of variation have a genetic or heritable basis to them. So these patterns become more complex as the organization of floral displays become more complex in itself. In my research, I went ahead and focused on, instead of focusing on a vertical single spike of flowers in plants, such as delphinium, which I had pictured previously, I'm going to go ahead and examine Visia americana, as it produces multiple branches each with their own display. So Visia americana is commonly known as the purple bench, and it is part of the pea family, and this plant produces a unique type of floral display. So here flowers are produced along the branches, and these flower-bearing branches extend upward toward the main stem. So as you can see here, you can see how each branch may have a same number of flowers or different flowers. It usually does vary between branches. It can go anywhere from 1 to 10 to anywhere in between. You can see how this organization stresses the possibility for a compound function value trait along the main stem and along these subsequent branches. So overall, my objectives here were to characterize the pattern for intra-individual floral variation in the purple bench, and also look at if these patterns experience any sort of natural selection. As mentioned previously, animal pollinated plants aim to attract pollinators for reproduction. Visia americana is visited by a number of a number of different species, but is typically pollinated by bumblebees. They are self-compatible, meaning that they have the ability to self-fertilize. For the attraction of pollinators, we went ahead and measured and associated 3 floral traits. So the first one's going to be B, which is going to be the banner. So this banner is associated with attraction for the pollinator for efficient pollen transfer. The FD is going to be the flower diameter, and FL is going to be the floral length, and these two are actually associated with the mechanical fit between the flower and the pollinator. So for our materials and method, floral traits were measured on all flowers across 40 individual plants. Each were between 5 meters apart, and this was done for spacing purposes, so that we can have unrelated individuals. So in that each individual flower or each individual would be distinct in themselves and not share any common parent. This project was performed in the month of June 2021. So there was a total of 757 flowers, and all successful fruits were collected, which was around 127, and the seeds counted was 453. So when collecting these data, they did go ahead and notate the flower position, branch number, and floral trait. So repeatedly produced flowers, as I mentioned, are not identical, but rather have this systematic pattern of variation, and this is all based on their flower traits, so the banner size, the flower length, and the flower diameter. This has a compound nature, as you can see here in plant 1539 and 1. This shows how they are not carbon copies, but despite this systematic pattern, and you can see right here, it differs among individual, and this can emphasize the compound function value trait between these three plants. So this compound function value trait creates a gradient within a gradient. There are patterns within the branches as well as along the branches. There are definite decline in the banner length and a lot of variation within that decline, as it can be seen here with the noise along this graph. And this noise is the effect of the different branches. So when you account for the branch, you get the noise on the left hand. And you can see how there is a decline in the differences in the pattern, but when you account for branch, there's an additional gradient that is accounted for inside the gradient. And now that I have investigated what they do and how these displays are affected by architectural effects and a compound nature, I will now move on to looking how these patterns experience selection. So phenotypical selection is usually estimated by using a traditional approach. So the Landian and Arnold equation is a linear regression of fitness on trait mean or a single measure. Here, the linear regression coefficient corresponds with beta, which is going to be our selection gradient. So this does show a straight regression using a response variable, the selection gradient, which is going to be, again, our regression coefficient, and an intercept. However, to account for function value trait, you really can't use this approach because function value traits are not linear. You actually do need to use a functional regression, and to use this, we use the selection on a functional value trait equation. This is a more appropriate method to measure selection on the gradients of a trait. So here is the functional regression will actually describe the gradient of trait variation, and it will use predictors as means instead of being limited, and while still accounting for branch number as well as the total number of flowers. So here, using this selection analysis, we did use a R package refund for the functional regression. And here the function, you could see it's going to be the function, which is going to be the fitness, which is going to be the seed set, is going to be the function, and it also does account for the brand number and total number of flowers. But as you can see, our function here does use our floral traits, so it's going to be our banner height, flower length, and width. Here we were able to select it to see how each individual trait measured, and it gave us our data. So looking at the selection in banner height, we can see how there was selection on the pollinator attraction was both within and along the main stem. There was a more significant selection detected on banner height than all three traits. So you can see on the vertical axis, it shows a partial regression coefficient across the flower position. And these lines then show how selection changes across the flower position. Selection was on both gradients. Here, selection becomes more negative. That is for like smaller banner height sizes on these subsequent branches. So here, the slopes and lines define the regression coefficient, And you can use this to kind of highlight the correlation between fitness and trait variation to show how selection can change over flower position. So here we did use relative flower number and absolute flower number, and this actually was necessary to include because the plants did vary in respect to how many flowers they did have. So by relative flower number, I do mean that flower 1 in this plant was compared to flower 1 in this one, regardless of how many flowers each one had. And as you go on, the last flower was compared with the last flower on the branch, just so that we can keep our data consistent, keeping in mind that each plant did have a different number of flowers on them. So in contrast to selection on pollinator attraction, in mechanical fit, which is going to be here, our flower length, selection was primarily along the main stem. So instead of 2 gradients, which was going to be the banner size, now this selection of gradient was only along 1. So similar to flower length, it's more consistent. Selection did not change as much for flower diameter, as it was also primarily along the main stem, and it exhibited main pattern along 1 gradient as well. So to conclude, I was able to see that and how Vicia americana displayed flowers as a compound function value trait. The traits that I associated with attraction, which was a banner, acted as a flag to attract pollinators. So the banner height was under selection in both gradients within and among branches, and the traits associated with mechanical fit being more morphological compatibility, seeing which pollinator can actually interact with the flower, was going to be the floral length and the floral diameter. And this was primarily along the main stem gradient. So as you could see in this video, pollinators do visit multiple flowers on the same plant, which could lead to selfing and inbreeding depression, on which one could associate this mechanism behind how our selection was detected. So to conclude, I give some acknowledgement. I do want to thank my advisor, Dr. Mason Kupaba, Lauren Sadwich, Nancy McFerrin, and Emma Collins. And I do want to thank you for listening to this talk.